Okay, so today we're going to be talking about cancer genetics, or in many ways you might consider it molecular biology of cancer. That is, the genes that are involved in inducing cancer, maintaining cancer, promoting cancer. We're not going to be talking about inheritance so much, but more about the genes that are kind of intimately involved in the process of tumorigenesis. And we'll be talking about two different types of genes. One are the oncogenes that I'll describe today, and hopefully in the next session we'll talk about another set of genes which are called tumor suppressor genes. So today I'm going to divide it up into a couple of different sections. In a sense, I'm going to give you a little bit of a background about some of the characteristics of cancer, and then I'm going to talk about the oncogenes, and the way I'm going to describe it is I'm going to kind of give you a history of how they were identified, because I think it's an important piece and helps us understand how important these genes are. Okay, so let's go ahead and start. We'll move to the next slide. So cancer, which is basically characterized by uncontrolled cell growth, which I'll kind of give you a few more characteristics in the next slide that's coming, but it's for all intents and purposes uncontrolled cell growth, and cancer is clearly considered a genetic disease. Not so much because it is inherited, because in most cases it's not an inherited syndrome, although there are a number of inherited predispositions to cancer. So breast and ovarian cancer is one. There's colon cancer, which is, there's a clear inherited predisposition. But you're not, in these cases, you're not inheriting the disease, you're actually inheriting a genetic predisposition which increases the likelihood that you'll get it. Cancer is considered a genetic disease, not because of the inheritance pattern, but because it has to do with mutations in genes that control cell division and apoptosis. Also, there's genes that control metabolism, and they control the immune system and cell migration. But the bottom line is that it's considered genetic because it results from a defect or a change in the behavior of certain genes and certain proteins. Now, these mutations occur due to many of the issues that we talked about earlier when we talked about mutagenesis: errors in DNA replication, environmental exposures, UV light, X-rays, chemicals in the environment, carcinogens, like I mentioned earlier, aflatoxin B1, or the benzoyl in cigarette smoke, so carcinogens that we ingest. Also, there's a number of plant compounds which turn out to be carcinogenic. Luckily, we don't eat an enormous amount of them, but basil, for example, which is a well-used spice, basil turns out to be there are carcinogenic compounds in basil. Also, there's a number of mushrooms that have carcinogenic compounds. There's in ferns, there's carcinogenic compounds, but we don't eat a lot of these, so in general they're not a problem. But the fact is that carcinogenic compounds are out in the environment, and if we ingest a lot of them, they can increase the risk of cancer because they will cause mutations. But I will say, and I think it's important to say, that aside from what is kind of talked about in the lay public, there is no dramatic, or there has been no dramatic increases in cancer incidence. There's not an epidemic in cancer. Cancer is not increasing in the population. In fact, it's stayed pretty stable. I think we're seeing more cases of cancer because people are living longer, the population is growing, so we see more cases. But for the most part, there has not been an increase in the incidence of cancer. There is no epidemic of cancer going on. And in the next few slides, I want to show you some of the evidence for that. So this is actually a very important series of data presented here. And this comes from the American Cancer Society, and these are studies that have been carried out. It's a little bit hard to see the print on this slide, but it's carried out between 1999 and 2013. And the different color graphs, different color lines represent different races. So African American is in red, Caucasians are in yellow, and there's islanders and so on, but the main point of this slide, what it's showing is all cancer sites, so we're not looking at specific cancers, but we're looking at the incidence, which means the number of cases per unit of population. And here it's the number of cases per 100,000 individuals, you can see on the side of the graph. And what's really clear is that if you look at all cancer sites combined, there is no increase in the rate of cancer in the population. And in some cases, it looks like it's going down. Like if you look at African Americans, the rate or the incidence of cancer has been decreasing over time. So unlike what people seem to talk about or what's in the popular press, there is no overall increase in cancer in the population on a population basis. But if you look at individual cancers, there may be a little bit of a difference, and we'll look at a few other slides. Let's take a look at the next one. So this slide shows a little bit different result, and this is looking at one specific type of cancer, and this is skin cancer. The previous looked at all cancers combined, and the rate actually is either stable or going down if you look at all. But if you look at, there are some cases, if you look at specific cancers, there have been some increases. So this shows male on the top, female on the bottom. This is the rate per 100,000, again, between 1999 and 2012, looking at the different races. And what we can see is that in both male and female, there have been some increases in the incidence of skin cancer in these populations, particularly in white, Caucasian, and Asian individuals, which is in green. So you can see the lines are, the slope is increasing, indicating that there is some increase in skin cancer, and this clearly has to do with sun exposure and not sufficient sunscreen protection and being out in the sun. So there is an increase in skin cancer due to lack of protection or more exposure to sunlight and UV light, which again causes damage to DNA. Let's go ahead and look at the next slide. Now, in this slide here, we're looking at one particular cancer type, and that's colon cancer. Now, this shows the incidence in colon cancer in two groups. One group is ages 50 to above, that's on the left-hand side. On the right-hand side, the younger people, ages 20 to 49, and what we see is dramatic differences between those two groups. In ages 50 and above, what we see in both men and women, there's been a dramatic decrease, significant decrease in the incidence of colon cancer in these populations. There's one very important reason for that, and that is colonoscopies. Colonoscopies are recommended for individuals who are 50 and above, going once every few years. Some say only once every 10 years, depending on whether or not they find polyps. But what happens is, when you have the colonoscopy, they search through the colon for what are called polyps, which are small growths, which in many cases can be precancerous. And so you do the colonoscopy, these polyps are identified and removed. Once they're removed, the chance of getting cancer drops to close to zero. So this is, in a sense, is a cancer which is absolutely preventable. Colonoscopy, remove the polyp, you're home free. Now, in the ages 20 to 49, there's been a very different observation, and that is what we're seeing is since 1994, there's a dramatic 51% increase in the incidence of colon cancer. Now, people between 20 and 49 are not recommended to get colonoscopies, although that will probably change. We have no clue. We do not understand the cause for this increased incidence. So there's people who are now studying it. In fact, here at USC, There's a number of groups who are working on this problem. Some think it has to do with diet. It may be, but there's clearly an increase in both men and women in the incidence of colon cancer. And the problem is that since people in their early 20s or 30s or even early 40s are not recommended to get colonoscopies, these things go undetected and then they end up developing the cancer. So those recommendations may change, but we need to understand what's happening there. So in addition to skin cancer, which has been increasing due to sun exposure, there is an unknown reason why colon cancer in younger groups is seen to be increasing. So let's move on to the next slide. So this slide shows a slightly different collection of data, and this has to do with causes of death. That is, what is causing death in the population. So we're looking at heart disease, we're looking at cerebral vascular diseases, which would be strokes and so on, pneumonia, influenza, and also cancer. And there's a couple things I want to point out here. First of all, in terms of treatments, we've been extremely successful with treating heart diseases and cerebral vascular diseases. That is, people have strokes and they can recover from those. Pneumonia, influenza, clearly doing much better with that. Heart disease has been very dramatic. The rate has dropped significantly between 1950 and 2000, as you can see on the very left-hand side. So in a number of different diseases, we've been very good at treating and preventing death. That is not the case with cancer. So there's two points I want to make about the cancer data. The first is that there is no dramatic increase in the death rate due to cancer. This is consistent with what I talked about earlier, that there's no increase in the incidence of cancer. On the flip side of that, we have not done very well at curing cancer. So people are dying at the same rate from cancer as they were in 1950. So with all the research that's gone in, and there's been a lot, and with all the drugs that have been identified, there still has not been a dramatic cure for cancer. Most of the time what happens is people go into remission and eventually it comes back. Or they can't be cured at all. But there has been no change in the rate of death due to cancer since 1950. Okay, let's move on. So what are some of the risk factors for cancer? What are some of the things that are thought to cause cancer? So this graph is a pie chart which basically breaks up all the various elements that are thought to be involved in cancer. So UV radiation is there, which of course would cause skin cancer, alcohol, viruses, family history. But there are two pieces which are the largest contributors to cancer, and these are things that we clearly can control. The first is that tobacco and smoking is responsible for 31% of all cancers. It's responsible for 90% of all lung cancers, but also contributes to other cancers as well. So tobacco and smoking is a huge chunk. And then diet and obesity is another 1/3 of the pie chart. And that is being extremely overweight, poor diet, meaning not enough fruits and vegetables, too many fats, not enough, not the proper nutrition, also can contribute to cancer. So there's two pieces that can be controlled in some way. And that is tobacco and smoking, and diet and obesity. The other things are a little bit harder to control. So let's look at some more of this. So here, in a sense, I've kind of simplified it. Call it what are the major causes of cancer, And I have three things identified. One is smoking. As I said, 90% of lung cancers, 30% of all cancers. Poor diet, carcinogenic foods, and then workplace contamination. It is working in an environment where there are pollutants that are produced, like alkylating agents, which can get into the air that you can breathe in, that can contaminate your lungs or your bloodstream. And so these three things are, in a sense, lifestyle. And that is, the incidence of cancer, in many ways, is thought to be able to be changed by changing of lifestyle. In other words, cut out smoking, you've got to cut out 30% of all cancers. So there comes the importance of what's called cancer prevention. And this falls into the area of public health. And there's individuals here at the Arnold School of Public Health who have been doing an enormous amount of research on cancer prevention. And many people, particularly those people who are working in that area, believe that the biggest advances that we will see in the future with respect to eliminating or reducing cancer in the population will not be from curing cancer. We've already seen that there's been no real impact on the statistics of curing cancer. Some cancers, of course, are cured, but overall, if you look at the number of deaths per cancer, we see that it hasn't changed. But what their thought is that cancer prevention, there was change of lifestyle, change of workplace, or cleaning up workplace, will have a much larger impact on reducing cancer in the population. And we've actually known this for a long, long time. And I want to give you one clear example, and this comes from an incident that took place a few hundred years ago, 1775 in London. And what I'm going to tell you about has become a classic case in public health and cancer prevention. The story goes like this: in London in the 1770s. First of all, London was a disaster in terms of cleanliness and hygiene. The streets were flooded with sewage, human waste. There was no good drainage. There was no flushing toilets. People would dump stuff out of windows. It was a pretty ugly place. But we're referring here particularly to chimney sweeps. In London, or in many places in Europe, in that time period, the chimneys were cleaned by what are called chimney sweeps. These are individuals that would climb up the chimney with brushes and clean the chimneys out, clean all the soot out. And because the chimneys were small, this tended to be young boys. Kids maybe 8, 9, 10, 11 years old would climb up and down these chimneys and clean them up. Now, in those days, as I mentioned, hygiene was really poor, and so people did not take baths. They didn't take showers, they didn't take baths. These kids would be up and down the chimneys, completely covered with the chimney soot, go home, go to sleep, get up in the morning, and just start again. What happened is, or what was observed was, there was a very high incidence of skin cancer in these chimney sweeps. Extremely high. And so one particular physician, his name was Percival Pott, was studying this and noticed that these boys had an extremely high incidence of cancer, and he made one recommendation. The recommendation was take a bath every day. Just get into the water and rinse yourself off. And they instituted that as a public policy, that all chimney sweeps needed to take baths after working, and the incidence of cancer basically disappeared. So this was the first clear example of public health policy that was able to eliminate cancer in these particular individuals. So that's why people think that if we can identify what the causes are, that cancer prevention will be a much more powerful way of reducing cancer in the population than curing it. So let's go ahead and move on and talk about some of the properties of cancer. So what are some of the properties of cancer cells? We know it's a disease, we know it kills people, we know it's uncontrolled cell growth, but there's many other properties that I want to describe. These are considered the 10 hallmarks of cancer that pretty much characterize properties of cancer cells. So I'll go through these one at a time. The first is autonomous growth signaling. That is, in normal cells, cells receive signals from the environment that induces them to undergo the cell cycle of cell division. So the signals come from the environment. What autonomous growth signaling implies is that cancer cells are producing their own growth factors that are stimulating themselves to divide. So it's autonomous growth signaling. They don't need an environmental signal. The second is no response to growth inhibitory signals. And that is, cells receive signals all the time in order to stop growing. You cut your finger, there's the healing process that goes on, the cells are dividing, recovering that tissue, and then at some point they stop growing because they receive signals that inhibit cell division. So cancer cells don't respond well to growth inhibitory signals. Third is they evade programmed cell death, and I've mentioned cell death, or I've mentioned apoptosis a few times, and that is in response to severe damage to the cell or severe damage to chromosomes. Often the cells decide to undergo this programmed cell death or suicide or apoptosis in order to eliminate these severely damaged cells from the population. And in many cases, cancer cells do not undergo apoptosis even if the DNA is severely damaged. Unlimited replication potential. This speaks, of course, to the Hayflick limit, and that is cells continue to divide indefinitely. They bypass that 50-generation Hayflick limit, and so they have unlimited replication potential, and that is part of the problem of cancer. The other thing is that many tumors will promote blood cell growth. And that is, if you have a solid tumor that's growing, the larger it gets, the harder it is for the cells on the interior of the tumor to get nutrients, because the diffusion only goes so far. And so many times these tumors will induce signals that allow blood vessels to grow and enter the tumor to feed the tumor to allow it to grow larger. And there have been some therapies, I'll just mention, there have been some therapies which are called inhibitors of hematopoiesis or angiogenic factors, which will inhibit the growth of blood vessels into the tumor and prevent the tumor from growing even larger. Another hallmark of cancer cells is that they have an abnormal metabolism. That is, they use an enormous amount of sugar in order to generate the requirements that they need to divide. They evade the immune system. That is, in many cases, the immune system ought to recognize that these cells are defective, and the tumor cells have ways of blocking the immune system or evading detection by the immune system. Genomic instability, and we've talked a little bit about that, and that is, if there's defects in DNA repair, then there's a higher mutation rate, and so these cells are constantly evolving, and that's why in many cases these cells will ultimately become resistant to chemotherapy because they're treated with these agents that will kill them. But then some cells survive because they've undergone additional mutations or changes in the genome that allow them to survive due to genomic instability. Many times they generate local inflammation, meaning that there's inflammatory cells that come into the area where the tumor is, and often the inflammatory cells are producing factors, growth factors, or other compounds that will induce cell division. And then finally, they exhibit mobility or invasiveness, meaning they become metastatic, and that is a serious problem because what that means is that cells, tumor cells that are in a particular location, now have gained the ability to escape from that tissue, let's say it's liver tissue, enter the bloodstream, travel to another site, let's say lung or brain, and then end up landing there and growing new tumors in those new sites. So these are the 10 hallmarks of cancer, which are considered to be the most important. Okay, so before we start talking about some of the genes that are involved in cancer, what I'd like to do is kind of give you a timeline of some of the more important events that took place in the kind of evolution of the field of cancer genetics. And that what are some of the experiments that led us to start understanding that genes were clearly involved in causing cancer, and so we considered it a genetic disease. It goes all the way back to 1911, and I go all the way up to the present. And it also shows how one generation of scientists and the experiments they do kind of spill into the next generation, and then there's a progression, and you can see how we begin to learn things. So the story goes back all the way to 1911. This is thought to be in many ways the origins of the field of cancer genetics. It goes back to an individual named Peyton Rous, who discovered what's called the Rous sarcoma virus, which is a tumor virus. And I'll tell you the story around that because it's kind of interesting. In the early 1900s, Peyton Rous was a physician, and he had a position at Columbia University in New York. And at that time, if you were a physician working at Columbia University, you were required to do rounds going to various farms, cattle ranches, chicken farms, in order to check on the animals. That was kind of their agreement with the community that's one of the things that they would do. So Peyton Rous would do his research, but then he would also go and do these rounds on these farms. And he went to one particular farm where there was a chicken farmer, and the chicken farmer told him that he had chickens with these extremely aggressive cancers, more than one, and it seemed to be spreading in his flock. So Peyton Rous was very interested in what the basis of this might be, and so he took a few of those chickens with these tumors back to his lab at Columbia, and what he did was he extracted the tumors, dissected them out, made extracts from these tumors, and then passed the extract through a variety of filters, so that in the final filtrate, the only thing that could be there, because the filter was so fine, were viruses. All bacteria and all the organelles would have been filtered out. And when he took these extracts and injected them into newborn chickens, what he found was that within 2 to 4 weeks, these chickens were dead from tumors which he labeled as sarcoma. So he had discovered some element unknown, which was extremely aggressive cancer-causing agent which these chickens were carrying. And because the filtrate would only carry viruses, he felt that there was a virus that was infecting these chickens and causing the cancer. He named the virus Rous sarcoma virus, and the implication was that viruses carry genetic information, although at that time, it was 40 years before the discovery of DNA, at that time it was known that viruses carry some kind of genetic information, and so his conclusion was that the viruses were supplying these cells with some kind of information that then was causing them to become tumorigenic. So some kind of genetic information was being passed, although they had no clue what this looked like. So that was 1911. In 1915, some more research being done by an individual, Yamigawa, in Japan. And what he was doing was he was looking at tumorigenic compounds that would cause tumors. He was using rabbits as a model system, and he was looking at coal tar. And this goes back, in a sense, 200 years to the discovery that Percival Pott made that the tar soot was probably causing cancer in these young boys that were doing the chimney sweeps. And so Yamagawa was trying to identify what was in that coal, that soot, which was causing the cancer, and he identified that it was coal tar, some kind of a substance that comes from the coal. Now, in the 1950s, so this was 40-some years later, the Millers was a group, husband and wife team of scientists working together. In fact, in those days, there were a number of husband and wife teams working together, but the Millers were chemists, and what they were doing is they were taking the coal tar and trying to purify compounds from the coal tar that would be causing cancer. So you can see the connection there. Yamagawa found that coal tar caused cancer, and the Millers in the 1950s were looking at what particular chemical compounds were in the coal tar that would cause cancer. And by the way, I actually knew the Millers. They were at the University of Wisconsin. I was at the University of Wisconsin in the late 1970s. This was, about 20 years after they did this original work, but they were still there, working together, still doing chemical carcinogenesis. So I would see them every day. But what they found was that there were carcinogens in the coal tar. In fact, I think they isolated benzoylpyrene from coal tar that would make these large DNA adducts that would mutate DNA. So here we go. Mutations in DNA are somehow connected to cancer. Now we move into the 1960s. Of course, there were still people like the Millers studying chemical carcinogens, but there were other groups of people looking at other characteristics, and one was that there was found in some cancers, particularly certain lymphomas and leukemias, that there were specific chromosomal translocations. We'll talk about chromosomal translocations a little bit later, but reproducible translocations, meaning that in specific tumor types, we would regularly see the same kind of translocation. So, for example, in chronic myelogenous leukemia, 90% of chronic myelogenous leukemia cases have translocations between chromosome 9 and chromosome 14, pointing again to some effect of genes or gene expression. Also, it was in the 1960s, it was found that UV light causes DNA damage, the pyrimidine dimers, and then it became clear later that UV light causes cancer. So again, it's connection to DNA. Now, in the mid-1970s or so, people started thinking, well, remember back in 1911, Rous discovered this virus, Rous sarcoma virus, that caused cancer? Let's look at that as a model system to see if we can figure out what's going on. Now we have new tools. We can clone DNA. We can start to sequence DNA. So it was discovered in 1970, which was 60 years after Rous first identified this virus, that Rous sarcoma virus carries a very particular gene, which was referred to as an oncogene. This was the first use of the term, and that gene was called SARC. So the virus induces cancer because it carries this particular gene. And then a few years later, in 1976, it was discovered that the SARC gene, which was responsible for causing cancer by Rous sarcoma virus, that gene, interestingly, was also present in normal cells. Even though normal cells carry this gene, they're not cancerous. So it's thought that somehow the virus was changing the gene a little bit, or maybe the gene expression. And that was referred to as a proto-oncogene, but I'll talk more about that later when we talk about some more specifics about oncogenes. Then in 1985, it was discovered that there were another class of genes in addition to the oncogenes, which are also intimately related with cancer, and those were called tumor suppressor genes. That is, genes that tend to be deleted or inactivated in cancers. That the inactivation of some function causes these cells to grow. And the first tumor suppressor gene that was identified was retinoblastoma, which is a gene which is responsible for tumors in the eye and the retina. P53 is another tumor suppressor gene. Then we move into the 2000s, and of course we have the first, with the Human Genome Project, the first complete sequence of the human genome. And so people started to look more carefully at the sequences and what these oncogenes and tumor suppressor genes were. And then in the 2000s, we started having identified what these genes are and then predicting the proteins that would be made by these genes. In the 2000s, people started looking at molecular modeling of oncoproteins and trying to see what they look like. And once we understood what they look like and what some of their activities would be, then the goal would be, or the goal is and still is, to, by molecular modeling, try and identify compounds or molecules that would bind to these oncoproteins and block their activity. And so in 2002, we have the first molecular targeted therapy, which was called Gleevec, which was specific to block the product of the translocation, which is seen in the chronic myelogenous leukemia. So Gleevec was the first molecularly modeled chemotherapeutic agent, very specific. And then now in the present, we're doing large-scale analysis of cancers, looking at hundreds and hundreds and hundreds of different cancer types, looking at all the breast cancers that we can, all the colon cancers that we can, all the lymphomas that we can, to try and identify the genetic differences between them. And so I think I mentioned very early in the semester that we see they fall into different classes, and that these different classes can be treated differently. So it speaks more to what we call personalized medicine. So that's about where we are now. Okay, as I'm working on these various slides, I'm realizing that the time is going by, and I don't know precisely how long we've gone for, but I think so far, well, this will be the 11th PowerPoint slide on this particular lecture, and so I'm afraid that it's close to an hour, and I don't really want to go that far, much longer. So what I said at the beginning is we're going to talk about oncogenes, but I'm going to change that. So this particular set of slides, this lecture really was talking about then the properties of cancer cells, some of the history involved, and some of the statistics of cancer incidence and so on. I'm going to close with this one particular slide, which basically introduces oncogenes and tumor suppressor genes. And then next time we will talk about the oncogenes, and then I think we'll have time also then to talk about the tumor suppressor genes. If not, then I'll do it the following week, which is fine, as long as I get to the material. So this will be the last slide for this set, and what I want to do is just briefly introduce the concept of oncogenes and tumor suppressor genes. And the bottom line is that many oncogenes and tumor suppressor genes, they're involved in controlling the cell cycle. And we talked a lot about the cell cycle early. We have the dividing cell, which receives, or we have a cell that receives a signal that sends the cells into S phase, and then into G2 in mitosis. And then if cells are dividing, you can also have cells that will receive a signal that will cause them to stop dividing, send them into what we call G0. So if we look, let's first look at the left-hand side, and this would refer to the tumor suppressor genes. So genes that control, in many cases, genes that control the entry into G0, that inhibit cell division, are referred to as tumor suppressor genes because the finding is they're inactivated in cancer. They're gone in cancer. So if that activity is gone, the cells do not respond to the signal to stop dividing, and so they continue to divide. It can either be stopping dividing or it can be apoptosis, some inhibition of cell division. So tumor suppressor genes are characterized by a loss of their activity in cancer cells. Now on the flip side of that, if we move more to the center of the diagram, we have cells in G0. They receive the signal and then enter the cell cycle, S phase, G2, and mitosis. And genes that control or promote cell division, those are the oncogenes. The oncogenes are genes that induce the cells to divide. And generally what we see in cancer cells are mutations that activate the protein. They activate or they stimulate the activity of that particular gene or that particular protein to lead the cells into S phase. So the oncogenes push the cells to divide, and the tumor suppressor genes would normally push the cells to stop dividing, but they're deleted in cancers or they're inactivated in cancers, and so the cells don't receive those signals. Okay, so I'm going to stop here. I want you to remember to take the quiz. Also, there's reading which will be assigned. I'll write it up on Blackboard. And maybe some questions from the book. I have to take a look at the book. Okay, so the next time, which will be Thursday, we'll look at the oncogenes. All right.